next speaker is Joe Crawford. He's a postdoc at the Andrew Leafy Group. He graduated from Washington State University in the Pillars. So welcome, Joe. Oh, there's a couple other things I forgot. Next week, the room was booked, so we will not have lunch with the Corps next week. The following week, our own Umnia is going to give a talk on our Steadicon system. So look forward for that. And then after that, Austin is going to give us a talk on X-ray CT, or X-ray CT system the week after that. So welcome, Joe.
Thank you. Yeah, thank you to the Corps facilities for giving me the opportunity to talk about my work that I've been doing here as a postdoc. So in today's talk, I'm going to give you a broad background about stomata's role in regulating plant water use and some of the challenges of measuring aperture. And as I'm looking out into the audience, it looks like there's a lot of plant biologists here, so it looks like I'm in good company. But then, I'm going to tell you about how I've overcome some technical challenges integrating confocal microscopy and leaf gas exchange. And lastly, I'll highlight some of the capabilities that we and you might be able to do with the new instruments that we're developing. So one of the main challenges that plants have is that they have to endure in place nearly constant variations in water availability, light, and temperature in both the short term and the long term, like seasonal and daily changes. And they have to do this all fixed in place in the soil. So during a growing season, plants need the sun to synthesize their sugars, but the sun is also the ultimate driver of evaporation of water in their leaves. And so water loss for a plant is something that really needs to be managed. And so one of the main adaptations plants have to regulate the rate of water evaporation and prevent dehydration of their leaves are these stomatal pores on their leaves. These are structures that can increase or decrease they can increase or decrease water loss from the leaf by opening and closing this central pore. Additionally, they have a role in altering temperature via evaporative cooling. And so these stomatal pores open and close in response to many environmental cues. So in this diagram, I'm showing how stomatal anatomy, environment work together to produce a minute-to-minute stomatal physiology. So stomatal anatomy, like size and shape on the leaf surface is primarily genetically determined. But the size of the pore opening is going to respond to several short-term environmental cues, short and long-term. So for example, generally stomata will open to light and close to darkness following the daily pattern of the sun. But you can also have things like this where you get some cloud cover and the central pore will shrink. So short-term drops in light intensity from either like leaf angle, cloud cover, all sorts of things. So the responsiveness of stomata to the environmental cues is necessary because it is thought that the photosynthesizing leaves are balancing the uptake of CO2 with the rate of water loss from their leaves. So stomata are also the opening through which leaves get CO2 for photosynthesis. So there's this dilemma that photosynthesizing leaves have to balance the uptake of CO2 and the loss of water. And this is particularly problematic because they share this anatomical pathway. You open up to get CO2, you're going to lose water. You get really hungry for CO2, you're going to lose a lot of water. At the leaf level, this can be described as moles of CO2 for moles of water. And for typical plants, somewhere around the range of 1 CO2 molecule that you gain for every 400 molecules that you lose. And so it's a big trade-off. And a related aspect of this struggle is that for most plants, CO2 is just passively diffusing inside to the leaf where biochemistry actually happens. And so if you start with a partial pressure in the atmosphere of CO2 at about 40 pascals, by the time you go through several cell walls, cell membranes, liquid phases, gas phases, by the time you're doing biochemistry, you're working with about half the concentration of what it is in the atmosphere. And relatively speaking, CO2 is actually very diffuse in the atmosphere. And so the end result is that CO2 is often limiting the rate of photosynthesis in many settings. Not all, but many settings. So you put that water for CO2 dilemma in a much broader context, and it makes perfect sense that water is the largest limitation to agriculture in the world, and that 70% of global freshwater use goes to agricultural irrigation. So to give a taste of the scale of what's going on here, there's some back of the envelope numbers to share with you. It's estimated that a single corn plant here will use about 125 liters per plant per season. So I like to imagine 125 of those camping Nalgene bottles. And if you're planting at 75,000 plants in a single hectare, that means you're going to transpire or use up 10 million liters of water in a hectare. Nobody knows what hectares are, so imagine an Olympic-sized track field with 4 Olympic swimming pools inside of it. That's about 4 Olympic pools per hectare. So the complexity of this problem has a lot of facets too, as there's an economic cost to pumping large volumes of water to remote rural locations. In drier climates, there's another facet that there's more evaporative demand from the atmosphere and from the soil. And so there's also temporal aspects, like here in the Midwest, we have the luxury of being supplied with ample rain, but there's also, it has to be raining at the right time, at the right place at the right time. And so there's a lot of facets here. So because stomata at all levels, from the cell to the ecosystem, are these gatekeepers of how plants interact with the environment, they're a key structure and system to study, measure, and understand for the potential benefit of agriculture. However, there's a technical limitation that stands in the way, and that methods for acquiring data about stomatal aperture are significantly limited. So some of the reasons that measuring stomatal aperture itself remains a challenge is that many of the methods used to do it are either unstable or imprecise. So for example, a common method is to use a special type of putty to make an impression of the leaf. So you'd apply this to the leaf, smash it down, and then pull it off and view it on the microscope later. So this can be successful, but often it is not. And it is at best a snapshot of stomata at the moment that you take the impression. And this is the best, by the way, this is the best image I've ever seen of a stomatal impression. So this is the putty went into the pore, and then when you pulled it out, it made this like negative of the shape. But at best, these things are imprecise and unstable. Another alternative is epidermal peels, where you actually will rip off the outer surface of the leaf and do a little bit of magic with buffers on it. And then, of course, you can just throw a live leaf under a microscope, but given all the environmental cues, it'll be closing in a few minutes and you won't be able to control what it's doing. So another way to study stomata and their responses is using gas exchange instruments. These are very popular with plant physiologists because gas exchange instruments can tightly control the conditions of a leaf cuvette to maintain or change the physiology of the leaf, even outdoors, as you see here. So, but it's important to understand how they work. And so as they measure a leaf area, they're measuring a flowing volume of air across that leaf area that reflects real-time stomatal characteristics, but these instruments are really defining a very important parameter that I'm going to keep saying throughout the talk. So I want to define it, and that's stomatal conductance. So what stomatal conductance is a measure of how easily CO2, water vapor, and other gases can be exchanged between the interior of the leaf and the atmosphere. So when stomata are wide open, there's little resistance to water vapor leaving, but when they're closed, there's a lot of resistance. And while this parameter is really accurate, I'm a big fan of it, it's important to know that this signal that you're measuring actually comes from several different types of sensors. And it's really think of it more of as a proxy for stomatal behavior rather than a perfect measurement of it. And so what are the real factors driving stomatal conductance? And I want to share this crude equation. So it's well established that stomatal conductance is a product or as a function of in the number of stomata per leaf area multiplied by by their stomatal complex size, and then their pore opening or their aperture. And so we know a lot about density and size thanks to a lot of new methods, especially the ones coming out from the Leakey lab. But the observed conductance, or what you would actually measure, depends on aperture. And this is variable and changing in time and space. And as I've shared in the previous slide, it can be very difficult to measure. So that means stomatal aperture at any given time is somewhere between 0 and 100% of its maximum value. And actually measuring that in realistic conditions remains a technical challenge. Also, it's highly influenced by the environment. So this inability to measure aperture means that a critical component of the overall phenotype is absent, and it means that there are significant gaps in our knowledge of how stomata behave in the real world. So my work has been to integrate simultaneous, non-destructive confocal microscopy with real-time measurements of gas exchange to get measurements of stomatal aperture under real conditions, and hopefully gaining biological insights into how these stomata are actually brokering plant-environment interactions. And so now I'll tell you about the work that we've been doing together with the core facilities on overcoming some technical challenges to do this. So our aim from the start was to visualize stomata under realistic, controlled conditions. And so what we did is we milled a custom chamber, and we put on a glass bottom, as you can see here. This is the custom chamber. Looks very similar to the standard. And we put on a glass bottom. And then we fastened the entire gas exchange instrument to a chassis that mounts onto the stage. So the entire the entire stage can move around and translate the entire machine. And so you actually can view all of the area of the leaf that is also being measured for gas exchange. And so at the start of this project, we had some hard goals, or uncompromising, things that we couldn't compromise on. One is that we had to have precise and accurate measurements relative to a standard gas exchange instrument. Secondly, we had to have tight control over realistic leaf conditions. And three, I wanted to do it without dyes and to be able to get a resolution and image quality good enough to measure an open pore. And lastly, I wanted it to be non-destructive. And so to test this instrument's accuracy, we took it to the greenhouse to compare what our custom chamber would do versus the standard manufacturer's setup. So we took data on the same leaf at the same time across several plants and leaf combinations to see if the data were comparable. And so in these figures here, what I'm showing you is the yellow line represents a one-to-one agreement between the two instruments. And so for the most part, these are raw values coming off of the instrument, or close to raw values. And so I think that we produce satisfying numbers to conclude that this is fairly precise and accurate for most needs. Now, to get images where our leaf would live a long and peaceful life afterwards, we need to have non-destructive imaging, preferably without dyes. And so, to my surprise, in part because I didn't know anything about microscopes at all, really, when I started this project, is that using plain old reflected light works really well thanks to the magic of confocal microscopy. So this is just reflected laser light, and it yields great images, and it's really great for repeated or long duration imaging. And then two-photon excitation really surprised me, also because I had no clue what it was or how it worked. But it seems to be exciting something within cell walls or cell membranes, and it produces a nice outline-like image that works well with post-processing. So lastly, we wanted to be able to image with really tight environmental control. And this is really something up to the LightCore, and it can already do this really pretty well. LightCore is the brand name of this gas exchange instrument. I should probably have said that, but it can all do these really well. So it's well known that stomata respond to CO2 concentration, and so in this experiment, it's really a demo, I raised the CO2 concentration to really high and really low, oscillating it back and forth, and then imaged throughout the whole time. And so in between the high and low CO2 times, there's only about 10 seconds where it's not at the CO2 concentration that I want it to be at, which is really good, really tight control. So both the images of the stomata and stomatal conductance as a readout are really corroborating each other really well here. So this is a GIF where it's, going back and forth, opening and closing, and you can see opening, closing, opening, closing, opening, closing, opening, closing. So really tight control. So to achieve these strict goals that we set out for ourselves of having tight control, yielding accurate, precise data, we ran into a couple challenges. And the first is that every time you change the geometry or the flow or add obstructions to these chambers, something like a big objective, it would really change the gas flow that's moving through it, and it really changes your data quality. And so the solution to this was to take long working distance objective lenses so that we can sit outside of the leaf cuvette entirely and just peer in. It was news to me that these long working distance objectives exist, and so I think it's really exciting that we could use it and that it actually works together with the Zeiss microscopes. And the second challenge is that pores are often very small, and so you have to have good enough magnification and good enough numerical aperture to actually resolve an open pore. And so we have 50x magnification and 0.5 numerical aperture. That's kind of like the resolving power. And so in order to get that distance sitting really close to the glass without breaking it, and I have broke it several times, we had to do a low profile chamber, but actually use our own thin foam gaskets and then change a lot of the sensors in their orientation so that they would be on top instead of on the bottom. And there's a couple recalculations you have to do, but for the most part, it's similar as before. And the last issue that we ran into is that vibrations due to the fans that are inside the leaf are really incompatible with confocal microscopy. And I was surprised to find out that these instruments have like 4, and unless you're really looking for them, you would only be able to find 1 or 2. So what we did, the solution to this was that we have a physical switch that we can turn on and off during imaging, and it can interact with the software to either lower the fan speed or turn it off. And this is part of a developing story about how to manage these vibrations really well. But you can see pretty clearly that the fan speed was a big problem that we had to overcome, because typically you would work at a fan speed of 100 or 80%, but the image quality is so bad that you're not going to be able to do that. So this is just quantifying what it looks like as you ramp up that fan speed. So now let's talk about some of the exciting applications of this equipment and touch on some of the research questions that may be possible now that we have it. So one of the applications of this tool is that we can really drill down and look at the movement of guard cells. These are the guard cells right here, for those not familiar. We can look at the movement of guard cells during closing and opening events. So in this video, I took Z-stacks, several slices of these, of a single stomata every 30 seconds, and then I collapsed each of those stacks and put it into a time course to look at the closure under a realistic environmental conditions. So these 3D views of the pore and the whole complex can really provide insight into the mechanical diversity of how stomata move. We can look under the hood and look at some of those structures moving. Because what's unique about stomata is that the way that these guard cells move is that they either take up water or expel water, and that's actually what causes the pore to move. But when they do this, they have to push on a larger hydrostatic pressure of the epidermis surrounding them. And so this is actually a big open question in stomatal biology right now, that this instrument will be able to, I believe it will be able to provide a lot of insight on how it's doing this. Another application is to look at dynamic environmental responses of stomata. So in this video, I took advantage of the fact that stomata change aperture in response to CO2 and light, and I captured the expected movements. So you can see the treatment that I did high CO2, low CO2, high, and then lights off completely, and you see the stomatal aperture tracks with the measurement of stomatal conductance quite nicely. So this is exciting because something as fundamental as CO2 sensing is really incompletely understood in the plant research community. And so one application would be to identify mutants of signaling pathways that could, figure out how plants are sensing CO2. Additionally, or optionally, you could add in GFP labels to really make this fly, or you could look at pathogens, stomata interactions on a single leaf in dynamic real time. So there's a lot of potential here. And I think another aspect that makes this really exciting is that a lot of people who have worked with the nuts and bolts of how guard cells open and close use some of those aperture measurements that I was talking about in the introduction, where they're really like violently ripping off the epidermis, putting it on buffer, and getting it to do things that may not be realistic. And so there's potential to test even a lot of the literature that's out there to see in realistic conditions if these channels and transporters do what we think that they're doing. And so I'm also trying to streamline going from image data to measurement data using machine learning. So with an example image like this, I've captured 4 stomatal complexes. And then you can see it in these masks. I've parsed these into different pieces like the pore area, the guard cells, the subsidiary cells, and then the complex as a whole. And then so once we feed that into a machine learning pipeline, we can get quick measurements going from image data to measurement data of the dimensions of these different components of the stomatal complex. And so in the rest of these panels, I'm showing you human measurements of these different components like stomatal complex versus a human-trained machine learning algorithm. And so you get really tight correspondence between these. And I think that once this is validated, what's exciting about this workflow once you go from image data, you can start to collect quantitative survey-like measurements of pores under some treatment. For example, high CO2, high light, low light, and then you can start to measure a distribution of stomatal pores. And another possibility is a treatment of reduced stomatal density. And so in my introduction, I mentioned how stomata in part control water use in an agricultural setting, and that stomatal density is one factor that could be manipulated to improve water use efficiency of crops. And so several groups all over the world, including my advisor, Andrew Leakey, and my colleagues here in the room, are transgenically reducing stomatal density in an effort to improve water use efficiency. The idea here being if these individual units are doing this exchange with in an intrinsic way, then reducing the overall density could limit water use without a large sacrifice in productivity. And so this tool that I've been presenting on today would be one way to validate that this strategy works, because it would be essential that there wouldn't be compensation of the pore, and so if it doesn't, it opens up even more fundamental questions that we could continue to ask. And so a survey measurement would look something like this, and as a disclaimer, this is preliminary data, so I might retract this slide in future presentations, but doing an experiment like this would be an example of what I was talking about, in that you could do a treatment like low CO2, ambient CO2, or basically low, medium, and high CO2. And the expectation is that stomatal conductance would decrease. And as I'm showing in these histograms, that's exactly what happens. So the measurement of stomatal conductance from the gas exchange instrument goes down, shifts to the left. And then you combine that with anatomical measurements like the area of the pore and the pore width, and you see a reflection of the same thing, and that the mean pore area is downshifting as well. And what's really key here is that you start thinking about stomatal apertures as a distribution or a population of pores, where some are wide open and others are very narrowly open. This isn't really news, but I think for me, it's a very different way of thinking about stomatal conductance, that it's a population of apertures. And before, you couldn't really measure aperture, but hopefully with this instrument, now you can. And so, in summary, I told you about how stomatal aperture dimensions govern leaf transpiration, and they'll vary with environmental conditions. I talked about how observing stomata while measuring gas exchange fluxes would allow advances in the understanding of leaf structure function relationships and leaf physiology. And lastly, that this equipment can dynamically control leaf conditions, measure stomatal aperture, and gas fluxes at the same time. I want to thank and acknowledge Dr. Andrew Leakey, my advisor or PI. I'd also like to acknowledge Dustin Mayfield Jones, who was a grad student who was working on this project before I was. And of course, everyone in the core facilities here, especially for inviting me to talk, but also because I bother them nearly every time I'm on the microscope. And so I think every time they see me, they're like, oh, there goes my day, because Joe's here and he's got a crazy whacked out idea for how he can make this better. But you know what? I'm really happy that they entertain that and are very nice about it, because I've been in other core facilities where they would say things like, oh, you again? That hurt. But anyways, I'd also like to thank Leakey Lab personnel, many of you, but especially Jeremy, and who's helped with a lot of the technical sensor and electronic things, taught me a lot about electronics. So with that, I'll be happy to entertain any questions if you have them, and thank you for coming.
comparing the standard to the custom one that you built, what were the different colors? Because you had a lot of points plotted on there.
Oh, yeah.
'Cause you seem to have like a little cluster of yellowish points that didn't really match. I was just wondering what the different colors were.
Yeah. The different colors in this case were indicating different, these were different plants and leaves. So in this case, what we were really trying to, when you do a comparison experiment like this, you have to have variation. And so we were going with like just the biological variation that was there in the greenhouse. And so these are different plants and leaves. Yeah.
Yeah, so how many stomates can you currently get in an FOV? And are you planning on measuring like the patchiness of stomatal response, like these guys grouped together respond differently than these guys, or stomates within a file respond differently than a file next door?
Yeah, so that's a good question. He was asking me at any given time how many stomata appear in a field of view under the standard setup. So under the standard setup, I typically get about 3 to 4 in there at a time. And then what I've done is I've defaulted to taking sort of a Z stack. So if there's any slope to the plane that I'm imaging, I capture it all by taking several slices. So yeah, it definitely, the potential is there to say like, okay, I'm imaging this stomata, I did a treatment, and now I'm going to move next door and see what they do. I currently can do that. I just haven't done that experiment. But what I can tell you is, you also alluded to this phenomenon of patchy stomatal conductance. This was a hypothesis that's been out in the literature for a really long time, and to be honest, I don't have a complete understanding of it, but it's this idea that if you have a leaf area of, say, this much, that you could have a patch over here that's really ripping and doing a lot of conductance, and then a patch over here that's doing hardly any. So basically saying you have spatial variation in how much vapor is coming out. And so what I have done is I've created like a net over the leaf area, and I hit like a grid. A grid is a better way of saying it. I make a grid, and I visit like all 20 of those spots. And I was really kind of surprised that there wasn't a lot of, it just appears to be random. So I haven't seen very much evidence of patchy stomatal conductance. But in that scheme, really, I mean, moving, I'm moving like 20,000 or 2,000 micrometers at a time. So it's like, it's not the next door neighbor. It's like the county over or the state over. Yeah. Yeah, same.
On the next, I think it's the next slide. No, the next one maybe. That one. So the noise, is that just from the mixing pan on the 60?
Yeah, so this is a product of that noise is mechanical noise from the fan. And so one strategy you can do, which I did here, just image through the noise and go slow, and that's one way you can do it. It kind of makes sort of a 1950s TV black and white image, but they were happy about it back then. I guess we could be. Yeah. Yeah, John.
I guess I'm just sort of thinking, both on that scatter plot that you had before, as well as like some of the applications. I guess I tend to think a lot from the opposite end of stomatal ontogeny, and I guess I'm like wondering now, between different genotypes, could the sort of functional lifespan of stomata also be variable? Like, could stomata sort of die at different rates?
Yeah, that was a sentence that I actually cut from my talk. But yeah, I mean, there's the possibility that you have 1,000 of these per millimeter squared, but only 500 are left living at any given time, or like they die or something, or become inactive. Or it's possible that they seal up in order to like cut their risk on fungal infection, because these are the gateways. This is a whole nother aspect, but these are the gateway to fungal infections or bacterial infections. So, yeah, it's possible that they're just sealed up and you're only using 50% at a time. Yeah.
That correlation plot, the diverse plant species?
All corn in this plot. Yeah, and part of that struggle was, or part of the selection of using corn is it's nice to have nice wide leaves. So one of the caveats here is that you have to fill up that entire chamber area. That's not necessarily true normally when using these instruments, but in my application, you have to fill up the entire chamber completely. And that actually reminds me that another change that I've made that didn't make it into the presentation is that typically when you measure with these instruments, you're measuring from both sides of the leaf. So you're getting really a signal from both sides of the leaf. But I've made modifications to trap the air on one side. So now that we're just measuring on the side that we're also viewing with the microscope. And that's been a cool thing as well. Yeah, Lynn.
Does this correlation include the on-off of the boundary fans, or is this assuming the boundary?
Yeah, well,
It's unfair because we talked about this late one night.
yeah, so this
Publicly.
this is like if you were to take it to the greenhouse and do your work as normal. So normal settings, mixing fan on and everything like that, yeah.
How much
So,
does your, whatever protocol you need to get to make the photos pretty, how much does that affect this correlation?
oh, I mean, if you were to do different mixing fan things on this, it would be wildly different. But what I did do is some modeling, and that what I did find, to my surprise, is that the mixing fan, you need some mixing fan thrust, but not a lot. It's sort of like an asymptotic thing. Like you need a certain amount, but after that, it's just excessive. Yeah.
And so how many feet of view do you need per plant? So how long it takes per feet of view?
That's a good question. So per field of view, including a Z-stack, it takes me about a minute to image, maybe in a minute and 30 seconds. And so, yeah, that's a really good question. Also did some modeling there. So like if you want to make an estimate of the mean and the mean and the error, how many stomata do you have to image to get like a good estimate of this? And so I did some modeling and basically concluded that you need about 20. So about 5 images would probably be enough to estimate your population of apertures and their variance, I think. Yeah.
You said the aperture size and the objective length was pretty crucial. Have you primarily done this on corn? Because I assume stomata size varies widely. Are there species and sizes, like tiny stomata, that will need more development? Or do you think this is robust for all species at this point?
Yeah, I think that corn is really nice to work with because it's somewhat rigid and big stomata. And I also use plants with like gigantic stomata, and those are great for visualization, but bad for physiology. I have taken some of those live live videos of soybean leaves closing, and they have significantly smaller stomata. It still works. You still capture it. It's a little bit fuzzy, but like it still works for sure. It's just not quite as clean. And it would totally work for if you're just trying to do treatment data, treatment data, treatment data. If you want to get like a live, sexy video, it's a little bit more challenging, yeah, because of the surface of the leaf and imaging of the planes and stuff like that. So, yeah, right.
What's you said for like a normal condition, if you wanted to measure it, it's like a 5-minute measuring thing. you talk about the live video, what's the difference? Like if you're measuring dynamic changes versus just pore size?
So that was about 1 minute for a field of view. And so if you want to do something like this, the strategy here is just to keep imaging in intervals. So just keep going.
So is this every minute?
I don't know what the interval is on that, but it's probably something like every minute. But I can say that if you're using the reflected laser light, it doesn't seem to have any physiological change on that individual stomata that you're looking at. So I've done that test, and it's pretty clean. So you can just, as long as the noise isn't an issue for you, you could totally collect continuous data on that.
But if you get rapid stomatal changes in several seconds, that's not something you can pick up yet?
No, I think that you could, as long as. Yeah, so the big sacrifice in time is taking the Z stack. But if you're just taking one image, it's like, you could do it as, really fast, like less than a second. The problem is that if you get any drift, you'll miss it. So it's a technical thing, but I'm pretty confident you could get like 1 second, 2 second twitches. Yeah.
Yeah, I'm just thinking this through. What's kind of the area for that image? Is it like 50 microns?
Yeah, I should have a scale bar in there.
I guess I'm just thinking, like, you could, I guess since the light core chamber is about 9 square centimeters, you actually have a lot of places that you can image without having to change the leaf. Is that right?
Without having to what?
Change the leaf in the chamber?
Yeah, I mean, you can cruise around with the stage and hit all sorts of things. That was my approach with the grid. I was like, okay, I'm going to visit these destinations in random order and see if there's a difference in aperture at these different places. And that turned out to be not very interesting, so there's a lot of work to do, and it was like, oh, pretty uniform.
You check that patchiness in corn, right?
Yeah, I looked at the patchiness in corn. It could be totally different in a different species, yeah. I think potentially a lot more interesting in other species.
That's really neat. Thank you.
Yeah, thank you. Yeah, Ryan.
Following up on your grid question, you said it's pretty much the same across, so I assume the way you clamp on one side of your chamber would be really close to the mid vein? So you're not seeing any differences the further away from the mid vein you get?
Yeah, that's a good question. So in corn leaves, you can kind of see here. There's this vein in the middle that is non-photosynthetic and doesn't have stomata on it. And Ryan is asking that if you get closer to that or further away, do you see a difference in aperture? And I think that you probably do. I don't have data for that yet because my grid is like not looking at the next door neighbor, it's looking like the county over, so, yeah. Is there, another question, okay.
Yeah, just a quick one, where I was actually kind of curious about the autofluorescence that you're sort of explaining for the two photon. I guess I'm just kind of curious, do you know, like offhand, like the excitation or emission that you're sort of using to produce those images? Like, I have a hunch of, like, what you're probably actually making light up.
Yeah,
Curiosity.
I should know this. I think Glenn Duncan, I think it's 780, it can change, but I think the 2 photon excites at 780, right?
Okay, tune it.
Yeah, I mean, you can tune it, but I think the,
780 is possible. You set it up, Duncan?
I think it was 7.
yeah. I know Austin was the originally who helped me set it up. He's not here today, but Yeah, I have a hunch that it's lignin that's lighting up. But I know we've talked about that before, and that it could be like waxes or other things, but my hunch is lignin. Yeah. Yeah, sure.
Does your custom head do fluorometry still, or could the microscope do fluorometry simultaneously with gas exchange and visualization?
Yeah, the custom head does not do fluorometry, but that is a very interesting idea that the microscope probably could somehow.
Have you done an emission spectra from your 2-photon? I thought we did that.
Yeah, we did do an emission spectra from the 2-photon, but I didn't want to include it in the talk today because it's kind of inconclusive, I thought, so.
So that would at least, that could suggest what you're looking at.
Yeah, but I think you probably recall, basically everything lights up at the majority of the spectra. There was really only on the extremes that it wasn't getting anything, so.
I forgot.
Yeah. All right, well, thank you very much for coming today.